Hi, everyone. Thank you so much for taking the time today to come listen to my research. It means a lot. So without further ado, I'll get right into it. So the title of my presentation here is The Power of Leaf Microbes, How Phyllosphere Microorganisms Can Help Protect Crops Against Drought. So my research is concerned with the intersection of two seemingly very distinct fields of science. So we have climate change in one hand, and we have microbiology in the other. So how do these two seemingly different fields connect? So we'll get right into it. So as you know, global climate change is becoming an increasingly prevalent issue throughout the world. It's leading to just a ton of different issues in different industries. And one of those industries is the agricultural industry. So a lot of crops that are important to agriculture are being negatively affected by climate change through different means. And one of those means is drought. So as global warming becomes more and more prevalent, um, different crops are exposed to higher levels of drought. So previous research indicates that microorganisms might actually um, be part of this a solution to protecting crops resist and recover from drought. Um, and specifically, my research is looking at forage grasses, which is a type of crop that's super important to agriculture because it's used to feed livestock. And right now, there's a current lack of knowledge on the bacteria that are present on the leaf surface, also known as the phyllosphere of these forage grasses. Currently, there's a lot of knowledge on the microorganisms that inhabit the roots of these forage grasses, and those microorganisms make up the rhizosphere. But there's still a lot of work to be done within the phyllosphere. So these phyllosphere bacteria, according to previous research, they provide drought tolerance through different processes, one of them being through potentially nitrogen fixation, through phytohormone production and biofilm formation. So those mechanisms are potential ways that these microbes might help their host plants survive in drought caused by climate change. So the purpose of my experiment is to explore how the phyllosphere of these forage grasses exist, and then better understand how the symbionts, these microbes, can affect plant growth in different growing conditions. And we also wanted to know what genes are present in the genomes of these microbes, so that we have better insight on how they can hopefully protect these plants against the negative effects of climate change. So we hypothesized that microbes in the phyllosphere possess mechanisms that can help forage grasses resist the effects of drought conditions. So to do that, we took samples from three different three different host plants, three forage grasses, these three right here. And we expose them to both drought and control conditions for the duration of 13 weeks. And we took samples once a week for those 13 weeks. And then from there, we isolated the DNA of the organisms present in these samples for gene marker analysis of the 16S rRNA gene and the NFIH gene, which is a gene involved in nitrogen fixation. So from there, we found the identity of our isolates, and then we looked for potentially beneficial mechanisms that could help our host plants. And then we performed some statistical analyses to find out what organisms were really present based on what sort of growing conditions they were exposed to. So up here we have our graph showing the relative abundance of different classes of species, or different classes, I should say, in the phyllosphere of one of our host plants, Lolium perenne that were exposed to drought conditions. So the drought conditions actually start here over in week 9. From the graph, you can see that there's an uptick of one class um, of Phyllosphere microbes, which is the alpha protea bacteria. There's a little spike here once the drought conditions um, are brought to the plants. So from there, we found that within that alpha protea bacteria class, Sphingomonas and Methylbacterium are genuses that are found within that class that are part of our core microbial community that we determined with our statistical analyses. We also found these genuses in our isolates. So, and then one of these Sphingomonas isolates have been found specifically to produce biofilms, which are important for protecting host plants from drought stress by retaining moisture on leaf surfaces. So that's the conclusions that we've made based on our results. But in the future, we hope that more research can be performed to better understand the mechanisms by which Sphingomonas creates biofilms. And we also want to know if there are other microbes that are present that can confer drought resistance and recovery to our host plants here. So I'd lastly like to thank Emily Bechtold and Klaus Neuschlein of the Neuschlein Lab for all their support. And I'd also like to thank Scott Auerbach for his guidance through the UMass Icons program. So thank you so much. Have a great day.